Okay.
Yes.
Oh, lovely.
There. Here they are. Can you see that, the presentation?
Yes, yes, thank you.
Okay. We are in. Thank you very much for your patience, and I apologize for this inconvenience. Everything okay?
Yes.
Okay. Well, first of all, I want to thank the opportunity to present this work that has been done in the laboratory of biophysics, in which we have been working during several years in trying to understand the role of water in biological systems. And specifically, we will try to summarize the work done by Dr. Rosa, the first author that has recently get his PhD degree, and also data of a graduate student, Jimena Cejas, that he will get his PhD degree in the next few months. My next collaborator is Dr. Frias, and I am trying to summarize all the beautiful things that they do in the lab, and I hope I can accomplish a good task. The aim of this talk is to try to demonstrate that water can act as a link between two theories of cells, the membrane and the colloidal theory of cells. Currently, there are two models for cell structure. One in which the membrane encloses an aqueous solution of proteins, the membrane theory, and another one, the colloidal theory, in which the cell is a crowded system containing gel of proteins embedded in water. In this last model, the proposal is that cell components are close enough and in fact, the cell components are in contact, having at most 2-3 water layers. So under this context, he thinks that it is appropriate to study the hydration properties of the membrane interfaces that may act as a link between the two approaches in order to compatibilize metabolic events with membrane events. In first place, what we need to define with some precision is what we interpret or we understand by the membrane-water interface. According to the Gibbs model, the interface is a plane, an ideal plane dividing the hydrocarbon region from the aqueous region, and no details of this interface is made. Guggenheim proposed that the interface is a region, a defined region of a defined thickness between the hydrocarbon region and the aqueous region, and this interface region is has very peculiar properties, very different from the two both solution in contact. This model can be extended easily to a membrane interface because we can design the membrane, or we can illustrate the membrane as a hydrocarbon regions and bulk water regions, and in between them an interface region that is mainly composed of the head groups of the phospholipids with its hydration state. Therefore, in this view, we can define the interface region as a binary, a bidimensional solution of hydrated head groups and water. And here is the first things that we need to clarify. which is the hydration layers that are in the hydration spheres of the phospholipid groups, and which are the region beyond, the water beyond that hydration layer. To do this study more clear, we can follow the dynamic aspect of lipid interfaces through the behavior of lipid monolayers spread on the air-water interface in a Langevin balance. As you may know, if we compress a monolayer, we decrease the area per lipid, and the surface pressure increases till it reaches a collapse pressure, in which the excluded volume of the head-curve region, that is in red, is surrounded only by a tight hydration layer, which is described in blue. In this collapse pressure, the surface tension of water nearly reaches to zero. So there is no excess of free energy, and in that case, it is important in order to have which is the thermodynamic answer that response that this membrane can have. When the collapse, when we have a surface pressure below the surface pressure, water confined between the lipids begin to appear, and therefore the surface tension begin to increase, and at very high areas, the surface pressure is at a very high surface pressure. The surface tension is near that of pure water. In surface pressure in between, we have a different surface tension, and if we, for instance, we choose a given surface pressure for a given area per lipid, we can have that, for instance, in a surface pressure of 20 millinewton per meter, we can have that the injection of a bioeffector underneath the monolayer, we have a response of the lipid membrane, and this response is proportional to the initial surface pressure. We can take the response of the difference between the final surface pressure in correspondence with the initial surface pressure, and we plot it in terms of the delta pi, in terms of the initial surface pressure, and we can see that the response decreases linearly with the initial surface pressure. And the extrapolation is that it's different for different lipids. We can have zero response at the surface pressure that is known as the cutoff. Therefore, we can analyze this plot also in terms of the water activity that implies the cutoff pressure. The cutoff pressure implies that there is a minimum of water activity that is needed beyond the hydration surface, the hydration layer, and beyond that, the membrane is able to respond to the stimulus of a bioeffect. The question now is, which are the properties that we have in this context? We can see, for instance, that this is the water activity corresponding to the collapse pressure, and this is the activity corresponding to the cutoff, i.e., the minimum water activity that the membrane needs to respond. And we see that the cutoff is a little bit higher than that of the saturation. So there is some kind of water that is beyond the hydration water that is needed for the thermodynamic response of the monolayers. The properties in this of this type of water can be studied by running the water balance in terms of temperature, and we can see is that in the presence of lipids, water presents a transition that is not seen in pure water. Here is the water balance in terms in the presence of lipids, that is the water balance in pure water. And this transition corresponds to the disruption of water tetramers, that is 4 H bonds water molecules, to monomers. And this transition takes place at the same temperature at which the lipid phase transition is observed. Therefore, the water in this case that is bound to the lipids acts as a mirror of the lipid behavior. Another properties that we can follow in this type of study, and in coincidence to what we have talked before, that there are very few water layers between the surfaces of membranes and proteins, is that there is possible a propagation between membrane and properties along the displacement of protons in the hydrogen bonds. And this displacement is can be explained the very important changes in polarization of confined water between membrane and proteins, and could be one of the reasons as the metabolic events or changes in proteins in the cytosol can be coordinated with membrane events and vice versa. In conclusion, we can say that the water excluded volume at lipid interfaces varies by surface pressure, osmosis, mechanical forces, and membrane components, in particular unsaturated lipids and cholesterol. That the excluded volume is related to the confined water between lipids. And finally, that this water ratio determines the membrane states that makes the membrane a responsive system interconnected metabolic processes with membrane events along H bond. So I want to emphasize the many things that have been said in some previous talks. For instance, the sensitivity to water to chemical environment is a very challenging idea to pursue in terms of to interpret which is the functional and thermodynamic role of water in cell behavior and cell function. Thank you very much for your attention.
Thank you very much, Professor Anibal. Now the floor is open for the questions. Any question or comment? Would like to have more discussion on this topic. Since the professor is present, it would be great to have a talk. Yelena, you have any comments?
Well, I love this presentation. Thank you very much. It is very brief, but this means really a lot. This is a huge work, and I'm really glad to see it this way. You closed. I'm sorry. I don't have a question, but I'm really happy to see because lately, for example, what we are seeing in our work, this interface behavior of water seems to be, for me, the most important part of water generally in the living cell. So, for example, if this water is lost, what would happen? Can you tell me that?
If you can recall I think the second or third slide, when we reach the collapse pressure, the collapse pressure means that the lipids are in contact between them with a tight hydration layer. This hydration layer is not able to respond. When the hydration, when there is water beyond this hydration layer, this water, yes, has an excess of free energy, and makes that the membrane can respond. What we don't know is how many waters are involved in that kind of second hydration layer that is able to respond. And that is a question that we don't have the answer yet. That's why I would be interested to explore more in near infrared, the water bands and all the domains, because we are talking about hydration layers or the water domains, and we don't know exactly which are. Not even the number of water molecules are there, and which are the structure. Because, for instance, we talk about surface tension, but mostly we talk about surface tension in bulk. But we don't know which is the meaning of surface tension when you have 10 water molecules, for instance, in between the lipids.
That's right. Yes.
This is a physical chemical challenge.
It is, but I think, especially with near infrared lately, I mean, now Professor Takeuchi is here, for example. So from his works, I started to let's say, understand what surface tension is in the terms of ratios of water molecular species that are present in the system, for example, that I'm examining. So it is about the balance between, let's say, hydrogen bonded and non-hydrogen bonded water. Maybe, I mean, he's here now, maybe he would, he can comment on that. But I'm also seeing in many other works also lately, and I can connect things like surface tension directly to the structure that I'm seeing on the aquagram. So I hope that we can actually, if you can have this instrument, we could then see entire picture in the terms of the our frequencies where this water species absorbs. So that would reveal much more, and we can confirm what you were actually are talking about today. Pierre has something to say. Pierre, you can
Okay. Professor Anibal, can you hear me?
Yes.
Okay, great, wonderful. So I was intrigued because this presentation you give is a classic example of how interfaces induce an ordering effect on the bulk phase of water. So there are two aspects. One is the dipole interaction because of water molecule ordering, which is more or less static, so to speak. However, there's also a dynamic aspect, which regards the oscillation properties of the polymers itself, the polymers being regular in structure. And I'm just looking at the screenshot that I made from your presentation. You have this hydrophobic head and this hydrophilic tails. They act like resonating antennas. So these, in case, could interact with the proteins in solution, with the water in solution. So there's a whole dynamic electromagnetic, let's say, ocean that modifies property, functionalities of these proteins. And also, as Gilbert Ling said, the induction association hypothesis, you need coherent water in order to activate a protein or to switch it off. You don't have to dissolve it, but you only need to block it, immobilize, like placing a corset around the protein, so it makes it silent. But however, if you induce decoherence in the water fraction, then suddenly the protein becomes again functional. So this is more or less my concept that I have in mind when I hear you talking, and I would see some more information on that. Maybe you have already some hints going in that direction that you have not mentioned because you're not so sure yet.
Excuse me, I couldn't understand the last part.
It's if you have already some hints, if this is already taking place, that water dynamics induced by the polymer on one side of the interface and the protein activity on the other, as Gilbert Link placed it, it's in his model, in his Association Induction Hypothesis, where he used ordered water in order to deactivate the protein and decoherent water in order to switch it on again. So it's a kind of proteomic activation. And you should be able to see this in your experiment. Have you any ideas? Have you any clues that this is happening?
Well, there are experiments that are not mine, but demonstrate that metabolic events across oscillation of in water structure can propagate to the membrane and induce, for instance, conformational changes in the membrane. What is not demonstrated is the other way around, how conformational changes in the membrane can induce changes in protein. What you talk about the dynamics, yes, it's true, and but we have to confine the dynamic of the process to very few water molecule layers. And what we are emphasizing in this presentation is that, for instance, we can see, for instance, the phase transition of a cell or in the membrane or even in the protein, we can see big changes in the polarization of water. But that changes in polarization of water doesn't mean that the molecule has to disrupt the hydrogen bonds, because a displacement of protons can give can invert the dipole moment of the water molecule. So that can be a reason of how propagation can be used along complex media such as a cell, and it's fully denied. Yes, in fact, there are people that are discussing about the fluctuation around some points in this regard.
Now you put me a flea in my ear, which is polarizability. This is very, very strong indicator from my understanding of the cell structure as the cell being a cavity resonator, which means there is an electromagnetic, let's say, cavity that has an intrinsic resonance frequency. And this should in turn affect not just polarizability, but also water cluster ordering within the cytosolic structure, along with the proteins, along with the organelles and all that stuff that's included in there.
Sure.
Reducing the degree of freedoms in the reaction so to become, let's say, a very specific functionality in the cell rather than being erratic and stochastic.
Yeah, I agree with that, but the problem is that we cannot in cells of a single water structure, because, for instance, we can have a different structure of water if you take, for instance, lipids and the head group of the lipids and the chain, hydrocarbon chain of the lipids. And we have a very complex surface, topologically speaking and thermodynamically speaking. And this is the unknown that we are trying to decipher, because probably in that arrangements, we have some codes in that the queue of the cell answer, even in complex systems, and the same in proteins.
This is interesting. Now, again, you bring sparkling ideas into my brain. It's what happens if you use a two-stage setup, one that involves a membrane associated into a protein soup, and you do experiments, and the other one you take out the membrane, the polymer, so to allow the proteins to act by themselves and to see if there's a change in reactivity in the protein dynamics. Would this be possible?
I think that there has been some some experiments that have been done with some lipids that can make a transition in terms of changes in protein structure, but they are not very clear. I think they are a little bit tricky, that experiment. And but if we have to compatibilize the two models, for instance, the cell as a compartment in which the cell interior is a broth of proteins in water, and the gel-like structure of the coacervate. This last one even has to take into account the presence of the membrane as a structure that may be involved in response of the cell to the environment. And in terms of that, I think the question of how this interfacial water can have a role in between, I think that is the question that we are trying to find.
Well, then you're on the perfect way to merge the membrane theory with the colloidal theory and put up a third theory. Can I summarize it that way?
Yeah.
What can be?
That in this approach that you're doing, you try to merge the membrane theory with the colloidal theory and come up with a third theory that encompasses both of these aspects.
Something else.
Yeah. Okay, well, I stop here. Well, anyway, thank you very much. Thank you very much.
Thank you very much. We have one more question for Professor Anibal from Sebastian Rosa. Sebastian, would you like to ask the question? So the question is, from which of the water band spectra part can you see the tetramer disruption and monomer formation? Sebastian, are you here?
Yes, I think that this question can be answered more precisely by Dr. Rosa, that I think is present because he did the thesis around that. It's a band around 360 nanometer, I think. 3060 nanometer, I think. Sebastian, are you there?
Hello, Anibal. I am here. I just asked the question because I wasn't sure if that was shown in the presentation, but mainly because when you see the water band, you can see like a broad band with different components between 4,000 and 3,000 wave number in the scale. So the question was more about the details between in which part was the monomer and in which part were the tetramer structures. And that was just in order for the audience just to know from which part of that spectra can you see the components. That was because when you see that broad band that is given for the hydrogen bonds of water, you can see the monomers at the higher frequencies, and you can see the higher coordination water at lower frequencies. And the answer is that you can see the monomer formation by the increase in the higher band population, and the tetramer disruption by the decrease in the lower part of that band. That is one of the components that you can find in that really broad band that gives water by infrared spectroscopy. I don't know if it was clear.
Okay, I think that is the technical answer of how we can show the tetramer disruption into monomer disruption is from the convolution of the broad band in the of what.
Yes, exactly. That was just.
I didn't have time to enter into details about that. But this is already published. Is this was published in the Journal of Physical Chemistry 2019.
I have a question. Can I ask something? Just say something. You mentioned about water activity.
Yeah.
And water activity related to free water molecules. I mean, this is the classical theory, and this is what people understand under water activity, right, at the moment.
Yeah.
So we have seen that lipids, in between lipids, we mostly have free water molecules. But water activity, Jelena mentioned, but water activity, recently we follow and we can see that it's not related. It is much more related to small water clusters. And what Pierre said, to me, it seems that it's more related to incoherent part.
Yeah. Yes. Incoherent.
Yes.
So-called incoherent.
Yes. So I think this is the key that we probably will have to concentrate. And if we have an infrared monitoring of dynamics of reactions, probably with the knowledge about water activity that we have at the moment. So we can make a step further in understanding of water structure in cell membrane. And what Pierre mentioned, and I agree absolutely, maybe that might bring absolutely new theory. It's not just A plus B, it will be something new, I think. But definitely related to water structuring and restructuring in response to coherence and incoherence or decoherence around protein. So we have water solvation shells around and hydration shell around proteins also. And obviously there is a communication. But this is something that we haven't proven and should go in this direction. But it is related to water activity, redefining water activity, I think.
Yeah. And I wanted to add what I saw in Professor Disalvo's work, the importance of these protons. This is what we also connected to water activity, that it is very, very fast water species, highly high energy water species. That is water activity, not just free water molecule.
No, I am not talking about free water molecules, or not completely free, because between the disruption from tetramers to monomers, we may have stages in which the water molecules may have 3, 2, or 1 hydrogen bonds. Yes. And that is a very complex landscape. In order to Romiana comment, water activity is a thermodynamic term, and thermodynamic acts in a number. And so we need to modelize which are the structural components that we can explain, by which we can explain the number. One of the things that has been done by the physical chemistry is the virial coefficients. And but now with some modelization of molecular dynamics, probably we have other tools to do that. But the question is how that water activity that is needed to the response of the bilayer which are the really structural sites
Yes.
or components that is like talking about in lipids, for instance, like in a specific site in an enzyme. We have a specific configuration in lipids that may have such kind of response, and how this is very complex. It's not a direct answer. But I think that with tools like near spectroscopy and these advances in polymers, in hydrophobicity and hydrophilicity, we can reach some answers in terms of to explore what is the meaning of the membrane interface in correlation to protein interface.
Yeah, I meant water activity. What we mean, we can measure water activity, and the basic for measurement of water activity at the moment is water vapor. Is vapor, right?
Exactly. Yeah.
So that's why. And this is, as you said, one number, but it's not that. It is not. It is more complex. And once we see this, once we are aware what activity means, then that will be one big step towards understanding of membrane and the functionality of membrane. I hope. Yeah.
Yeah.
And exactly what Pierre said. So we are on the way to see, probably that will open also and answer the question about coherence and decoherence.
Yeah.
So I hope so.
Yeah, well, we have to try.
Yes. Thank you, Anibal.
Thank you to you.
Thank you, Professor Anibal. So this was our last presentation. I'm not sure whether Felix is here or not. Felix, are you here? We really enjoyed your presentation. Would you like to say hi to all of us? We have a question for Felix. If Felix is here, he can answer that question. Oh, so Felix is gone now.
He doesn't want to hang out. Maybe he had problems connecting. I'm not sure.
Oh, okay.
But he was in the meantime. Anyway, I just wanted to comment, because I know that you gave word to Felix. I'm sorry. I just wanted to comment a little bit more on Professor Disalvo. Really, I'm very, very grateful for you that you made this talk and were available here for questions. This was really beautiful to attend.
Yeah, we are thankful.
Thank you to you.
And I wanted to say, you did a huge sacrifice, because in Argentina, it is now very early in the morning. So I think the whole audience is really grateful that you did that for us. Thank you.
Well, I hope that the talk was useful for the purpose of the meeting.
It was absolute purpose. And I'm glad to see the whole paper published about this. And that we had this paper in our aquaphotomics special issue.
May I have an announcement, if you allow me?
Absolutely.
Yes.
Next August in 2023, in August, 3-4 August, I think, we are trying to make a conference about frontiers in biophysics, in cell biophysics, in which we are trying to collect all the sciences that are coming from quasar-based theory or membrane theory, water theory, water participation in cells and in tissues and so on. And we are doing that conference in memory of one of our colleagues that passed away last year, the Dr. Bagatoli, that tried to compatibilize the lipid response with the link theory in terms of which are the oscillations and this dynamic aspect that was mentioned. So I will keep I should like that you have it present for the next year, and through Romiana or maybe you, Jelena, can help us to make the diffusion of this event.
Absolutely. We will. we will notify all our society. It would be wonderful to attend. Thank you for saying this here.
Okay.
It's a great contribution, and I think the idea of having this conference is really great. So we'll try to contribute as much as we can also.
We hope to see you in Argentina again, Romina. And you too, Philippe.
Yes.
I would love to come. I've never been in South America. That would be my first time.
Activity.
I have to work on water activity. Yes. You have a good contribution.
Okay.
Thank you very much.
Thank you.
To all of you.
Thank you very much to all of you.
Thank you very much, Professor Anibal. Professor Senkova, would you like to talk about today's webinar, about 4 different presentations?
Yeah, I was listening.
Any
And
comment?
I was thinking that 4 different areas, and we could see absolutely new venues. And what's better than this? Thank you very much to all people attending, and I'm so happy that we see more and more people and new people coming. So if we could have more and more sharing, and our YouTube is visited by many people also. So thank you very much to everybody contributing to this. Jelena, thank you very much. Also, Harpreet, thank you very much. So we continue with we're a little bit behind the schedule, but because always there is lots of work. But Jelena keeps on track very strictly. So I hope to the new webinars are coming with new papers and new topics. So it's getting warm, interesting, and thrilling. So
Yeah.
thank you.
Yeah, Jelena, would you like to add a few sentences, 1 or 2?
Harpreet, I'm very grateful. You moderated very nicely the entire session, and I'm glad that I was relieved of this responsibility to, always monitor. Oh, did I forget this question? So it is hard for me when I moderate to listen actually to the talks. So I'm very happy that you can join us and help with this. And it is good to have new faces.
It is my pleasure. It is my pleasure. Yeah, I enjoyed being a moderator.
I really appreciate this, how to say, new idea. And it will be so good to have people joining Jelena in leading the webinar. It will be very nice.
And especially, I mean, it's not only me. Harpreet is so long in aquaphotomics. Why not?
Yeah, my PhD was in aquaphotomics. Yes, I enjoyed doing aquaphotomics, and there is more to learn about aquaphotomics.
Yes,
I would say.
Just started.
Yeah, thank you so much. Thank you, everyone. So we really enjoyed our 4 presenters and their presentations. If you have more questions, you can type your questions in the chat box or send an email to either Jelena or Lawrence.
Yes, to aquaphotomics.
Yeah, to the Aquaphotomics Society. Yeah. Yeah, and I also like to thank our foundation, Tsukino Shizuko Foundation, and about the recording of the presentation of this webinar will be available in a few days, and it will be sent to you or shared on aquaphotomics.com website. So thank you very much.
Thanks to Lawrence. Thank you, Lawrence, too.
Yes, Lawrence is actually the most here responsible, I guess. Cheers to Lawrence.
Thank you very much.
Yes, thank you.
Thank you all.
One news is last maybe we're having the Japanese aquaphotomics conference this month on 23rd and 24th of November. The place is Yunosato Onsen in Hashimoto City in Wakayama. Most probably we'll also, you can view the talks. We'll try to do this to see if we can organize this, to see the talks on our YouTube channel.
Online.
Yeah.
Yes, I would like to see the talk online.
It will be Japanese though, but
Oh.
so
So there are no
will be.
English subtitles?
Oh, we could do that. We should.
no, well,
We should.
good idea. Yes. So and many events this month, so busy as usual. But thank you very much for to everybody for attending the webinar.
Bye bye. Thank you.
Bye.
Bye bye.
Thank you.
Bye. Thank you
It's
very
always
much.
the hardest thing to finish.
Yes. To say goodbye.
So
Absolutely.
goodbye, everyone. Thank you.
And see you in the next
Bye-bye.
webinar.
See you next time.
はい、内田先生。
Thank you.
青細田先生。
Thank you.